This is the supplemental lecture titled Cell Membrane and the Cell, with a focus on cells as the living units. So the membrane is the barrier between the intracellular and extracellular environment. The internal environment, which they compose, is divided into two spaces, that intracellular and extracellular, and of course they are separated by the cytoplasmic membrane. For physiological function, especially relative to homeostasis, this becomes extremely important because we have to have a balance between the external and the internal environments, and of course the internal environment, environmental balance, is the focus of this component of this lecture. This slide illustrates this system that we're talking about, with the external environment and the internal environment and a barrier in between. So some cells are protective cells between the internal and the external environment, and some are exchange cells such that materials enter and leave the body. Within the internal environment, of course, there is the extracellular fluid of the internal environment and the intracellular fluid of most cells. And so we see there is movement into and out of cells, into the and out of the extracellular fluid compartment, and again with the extracellular fluid compartment through the cellular barriers to and from the external environment. These are very important concepts for understanding physiology of organisms. This slide is showing us the distribution of the fluids within the internal environment, and how the cell membranes serve as barriers between those. So here we are seeing the blood within a capillary or blood vessel, with the extracellular fluid component being the plasma, and the interstitial fluid within the interstitial compartment, a subset of the extracellular fluid compartment. So the interstitial fluid and the plasma are both components of the extracellular fluid compartment, two subsets. And then with the tissue cells, we see the cell membrane serving as the barrier between the interstitial fluid and the intracellular fluid. So plasma plus interstitial fluid equals the extracellular fluid, with the cell membrane serving as a barrier to separate that from the intracellular fluid. So this slide is demonstrating that the plasma membranes regulate fluid movements from one compartment to another. So we begin that concept evaluation by looking at the formula: total body fluid equals intracellular plus extracellular fluid. So here we see a blood vessel wall, a capillary wall, with a tissue cell outside of that. So we have the interstitial fluid, interstitial space with the interstitial fluid as a compartment, separated from the plasma of the extracellular fluid compartment by cells, the endothelial cells. So there's the membrane barrier there that we saw on the previous slide. And the general somatic body cells we see with their cell membrane separating intracellular from interstitial. So the question is, what is another name for intracellular fluid? Of course, that is cytoplasm. So the balance between the cytoplasm and the interstitial fluid compartments is extremely important in physiological studies and for understanding physiology altogether. So the membrane that we're discussing here is comprised of a lipid bilayer that contains proteins attached or inserted within it. The lipid bilayer, of course, is lipid soluble. It has phospholipids and cholesterol. The polar head of the phospholipid has a charge, therefore it has a physical property of being partially hydrophilic, and the hydrophobic or lipophilic environment is created by the legs of this that extend from the head of these phospholipids. Proteins are of two types. They can either extend through the membrane, so spans the entire width of the membrane. So we'll have transmembrane spanning segments. And then the peripheral proteins will be only partially spanning, so they will be inserted into the membrane only, we'll say halfway, but it might be more than half or less than half. Thus, with the integral proteins, we create selective permeability, and if it's clustered with other integral proteins, it might form a channel, and we would see movement of substances across the membrane as well as transduction of signals, so transmembrane signal transduction. And, of course, it would function as ion channels, one of the most common type of channel that we would see. There are other examples of how these proteins work, but we will start with these. Peripheral proteins are exposed either to inside or outside, but they don't span the membrane. So typically they're not going to be transporting molecules, and it will function as a binding site for a receptor, or it might be some type of a second messenger. It has two examples. So here we see the composite cell with the cytoplasmic membrane around the outside here. We see microvilli increasing the surface area, and deep within is the nucleus, and surrounding the nucleus is the cytoplasm. So the cytoplasm actually has a fluid component called the cytosol, to be more specific in the answer to the previous question on the previous slide. If we start with the nucleus, we can look at its makeup. There's the chromatin with the DNA and proteins, and then there is, of course, a fluid compartment within the nucleus as well, because it is an aqueous medium. And the nuclear envelope is two lipid bilayers, and there are nuclear pores for transport movement into and out of the nucleus. There'll be more details on this in a further lecture. So the cytosol of the cytoplasm will be surrounding all of these membrane-bound organelles. We, of course, see the endoplasmic reticulum, the rough endoplasmic reticulum, the smooth endoplasmic reticulum. Here we see the Golgi apparatus with some vesicles for secretion, and the mitochondria, the centriole with the cytoskeleton being illustrated as well. There will be more detail of this in a future lecture. So if we focus on any of those membranes that we saw, the protein content will differ. The specific types of phospholipids, the preponderance of one over another, will differ as well, but this basic structure will be the same. So here we see the lipid bilayer. These are the polar heads with the tails or legs coming off. On one side of the lipid bilayer membrane, and on the intracellular side, the extracellular side, polar heads are exposed as well. So it tolerates an aqueous medium on each side where we have the polar heads. We'll look at this arrangement of these molecules in a bit more detail in a moment. We see examples of integral proteins scattered throughout here, and we'll talk about more examples in just a moment. These yellow molecules are cholesterol, so they are actually inserted within the membrane. So because these proteins span the membrane, they have a transmembrane spanning portion, they are integral. We notice on this integral protein, there's a peripheral protein sitting on its outer surface, and on others, there are some sitting on the inner portion. These would be considered as the peripheral proteins. So the peripheral proteins on the inside might attach to cytoskeletal elements, or the cytoskeletal elements will attach directly to integral proteins. This is functionally important for the shape of the cell and moving these proteins within the membrane, and we'll discuss that in a bit more detail as well. So first, let's consider some examples of these membrane proteins. So these are integral proteins, this one obviously forming a channel. Some might require energy in the form of adenosine triphosphate, or ATP, to function as a transporter. So the channel protein labeled A, it's a protein that spans the membrane, may provide a hydrophilic channel across the membrane that is selective to a particular solute. So it contributes selective permeability to the membrane. First, it's the route for water-soluble molecules. There is no selectivity for lipid-soluble molecules because they simply diffuse through the membrane. So selective semi-permeability, first selective for water-soluble or hydrophilic, and then it'll even be selective for a particular ion or particle that would be dissolved in the aqueous solution, so therefore that particle would be a solute. Example B, some transport proteins will hydrolyze ATP as an energy source to actively pump substances through the membrane. Other integral proteins might simply have an enzyme function internally, as we see here. So it's built into the membrane, can be an enzyme with an active site exposed to substances on the in the adjacent solution. So in some cases, several enzymes in a membrane will act as a team that catalyzes sequential steps of a metabolic pathway, as shown here, from right to left. It doesn't seem to go from right to left, does it? Oh, yes, it is. Here's the right, and we're progressing to the left. So it binds here. The substrate binds to the enzyme. The product is here. That is the substrate for the next enzyme in this sequence. And here's the final product. Another example, receptors for signal transduction. So a membrane protein exposed to the outside of the cell may have a binding site with a specific shape that fits the shape of the chemical messenger, such as a hormone. The external signal may cause a conformational change in the protein that initiates a chain of chemical reactions in the cell. That, of course, that type of signaling will be studied in greater detail later. We might see intercellular joining, so a cell-to-cell junction or intercellular junction. So the membrane proteins of adjacent cells, integral, therefore exposed outside and inside of the membrane, can hook together in various kinds of intercellular junctions. Some of these, like cell adhesion molecules, provide temporary binding sites that guide cell migration and other cell-to-cell interactions. There might be cell-cell recognition mediated through these proteins. So one cell membrane with a certain type of protein here, serving one part of the recognition aspect of recognizing, and this cell membrane protein providing the signal to be recognized. In addition, we know that cells need to stick to the extracellular matrix where they reside. So here we see an example of the lipid bilayer membrane with an integral protein and the peripheral protein on it that binds some of the cytoskeleton inside the cell. Outside of the cell are extracellular matrix proteins that typically have these long branch chains of complex carbohydrates, and they are called extracellular matrix proteins or fibers or fibrils, depending upon their size. And elements of the cytoskeleton, the cell's internal supports, and the extracellular matrix or ECM may be anchored to the membrane proteins, which help maintain the cell shape and for the location of certain membrane proteins. Others play a role in the cell movement or bind adjacent cells together. So some of these proteins in the membrane might have these complex branch carbohydrates on them as well. The binding of a carbohydrate to a protein, of course, produces a molecule called a glycoprotein. So they are commonly found in the membrane. Virus membranes have many of these. They are potent antigens, so producing a vaccine against a virus. Typically, it is targeting some type of glycoprotein on the outside of the virus, or it can be a potent immunogen. I know these seem very much the same. An antigen is an immunogen, but typically the immunogen we're thinking of being exposed to it rather than receiving a vaccine. It's the point being made here. So they serve as binding sites for receptors most commonly. That carbohydrate can be involved in the molecular recognition and interaction between the signal molecule and the receiving molecule. The fluid mosaic model is very important for understanding movement of proteins within the membrane. And this is basically what's being emphasized here, but keep in mind that the phospholipids can move around too. There's some evidence that phospholipids will even flip from inside to outside layer of the membrane. So it isn't a rigid, fixed structure. Things can move around within it. Excess cholesterol in the lipid bilayer will decrease the fluidity of the membrane. Ethanol will increase the fluidity. So this is a figure that will show us some of the events related to the membrane cycle within a cell. And I'll take a moment just to get some orientation here. With the key, we see that we're talking about receptor-mediated endocytosis for the importation of materials that are needed by cells. So this is a discussion of receptor-mediated endocytosis. So at this point here, number one, we see binding occurring in the extracellular fluid, the interstitial fluid. So a receptor ligand complex is produced on the surface of the cell. The ligand is the molecule that binds to the receptor on the surface of the cell. Where the fluid mosaic model becomes important is that there is sequestration of these ligated receptors, and we see that shown here. And then this sequestration of the ligated receptors triggers binding of protein on the inner surface of these, protein called clathrin, and it indents a little bit, forming a pit, and this is referred to as a clathrin-coated pit, to the point where this pit deepens more and more. Any change in shape or movement of a cell or within a cell is related to an interaction with the cytoskeleton. So we have to envision cytoskeletal proteins attaching to this membrane, the clathrin very likely being involved in that interaction. We go to step number 2 here, extending the discussion from this point where we have an invaginated plasma membrane. We now have it breaking off and floating into the cytoplasm, not really floating, cytoskeleton pulling it in. So we have a vesicle formation occurring here. And we now have a clathrin-coated vesicle. The pit becomes the vesicle. And now we see there is an uncoating, so the clathrin will be freed. We see the clathrin freeing up here to go back in and be available for reuse. We now have an uncoated vesicle. Just orienting my slide. So the uncoated vesicle goes through step 4, which is a fusion with an endosome, an early endosome. And we see many different types of events occurring here, and we can have recycling of receptors to the plasma membranes. It is really recycling of membrane as well. Or there can be a carrier vesicle that will bring some of the contents of this endocytosed structure to a late endosome, and that late endosome has a pathway. But first, let's follow this carrier vesicle for recycling of the receptors, step number 5, delivering them back to the membrane for reuse. That's very important in pharmacology to understand receptor downregulation. It's a very important key aspect of desensitization and actual downregulation of receptors in cells. For our point, we're just seeing this fluid mosaic model and membrane recycling or cycling concept. So number 7 here, there can be transcytosis, very important for endothelial cells to transport particulate matter from the plasma fluid subcompartment to the interstitial fluid subcompartment on the other side. So the carrier vesicle will fuse to the inside of the opposite membrane of the cell and then open up and exocytose what was endocytosed through receptor-mediated endocytosis on the opposite side. The alternative, number 6, we did number 7. We did number 7 here for the exocytosis. Number 6 is degradation in a lysosome. So it'll fuse with a lysosome, and we see degradation of the entire contents. So let's now talk about steroid and certain drugs, or the permeability of the membrane, and just get through some basic concepts. So lipid-soluble steroids and other drugs can zip through the lipophilic environment of the membrane. And this is dependent upon concentration gradient. So we always move from higher concentration to lower concentration, whether we're talking about solute, which is lipid or water-soluble, or if we're talking about osmosis, the movement of water is always, the water diffuses from an area of high water concentration to low water concentration. It's opposite of the concentration gradient of the solute, but the solvent is going from its higher concentration, pure water, let's say, to one that has a lower concentration of water, say salt water. So it's a very, this concentration gradient is a very powerful driving force for the movement of molecules, whether we're talking about solvents or solutes. Unless we use energy to push it in the other direction, of course. So the membrane receptors, there can be 2 types, metabotropic, so it'll change the cell's metabolism when it is transducing a signal across the membrane. So this transmembrane signal transduction, or ionotropic, so it'll allow ion movement. So we'll see more and more examples of this when we study neurophysiology. Now the point of selectivity, after semipermeability, there's selectivity. So here's an example. If 2 amino acid residues that are specific for calcium are present, calcium passes through the channels when they open. So that transmembrane spanning segment of these integral proteins can have certain amino acids present there, and certain features of these amino acids are turned and presented within the channel, and they create an environment that is selective for calcium. So 2 amino acid residues is sufficient. So what kind of drug is being used that affects channels to treat for hypertension? Of course, calcium channel blockers. So we're able to exploit that. So let's take a peek at that concept and see what we can gain from it. So we're looking here at an ionotropic membrane receptor. It is a ligand-dependent ion channel, transmembrane spanning ion channel, and it is selective, but it doesn't depend on energy. It depends upon a ligand. So in the unligated state, the receptor, it has a structural conformation where its channel is closed. In the ligated state, when the ligand, the extracellular chemical molecule signal, binds to its binding site on the extracellular portion of this transmembrane spanning protein or integral protein. It triggers a conformational change such that the ion channel is open, and then the ions will move down their concentration gradient. So if we are saying this is outside the cell and this is inside the cell, obviously illustratively we've got a concentration of 4 outside, 0 inside. Our concentration gradient is outside to inside. An example of a metabotropic receptor is illustrated here. This is the type related to generation of cyclic AMP as an intracellular chemical messenger. So we start first with the extracellular, intercellular, between cells, chemical messenger that ultimately will be transduced to this intracellular second messenger. So this is the first messenger, extracellular, intercellular, between cells, will bind to its receptor. The ligated receptor now undergoes a conformational change, triggering the activation of an enzyme, adenylate cyclase, that acts on ATP to cyclize it. Here we see adenosine triphosphate, the 3 phosphates. Adenylene cyclase cyclizes the molecule, takes off two phosphate groups, so it's AMP, monophosphate, and that phosphate links this carbon to this oxygen here and cyclizes it. This now will have a receptor, binds to it, and will change the conformation of that. Here we see an example of a protein kinase that is inactive. The cyclic AMP binds to it and activates it. So we now have a protein kinase that is active. Kinases, by name, are enzymes that phosphorylate other molecules, especially proteins. And when certain enzymes are inactive and they are phosphorylated, they become active, then it will produce, if it is a rate-limiting reaction, then, and that enzyme is key to it, then you can have significant, this isn't the only example, but it's the one shown here, you can have significant physiological effects of the hormone within the cell. This is metabolism, so this is a metabotropic type of receptor. We haven't simply changed the ionic flow across the membrane or the intracellular ionic concentration. We have actually transduced into a change in metabolism. So let's spend a bit of time looking just at this lipid bilayer and its semi-permeable or selective permeability. So again, we first orient to the concentration gradient. So we have concentrations outside the cell, concentration inside the cell. So the direction of the concentration gradient force, the potential energy is directed inward. We have a certain membrane thickness, and that thickness is made up of the phospholipids on each side, and then the composition of the lipid layer forming the lipophilic or hydrophobic portion is in the center of this two-leaflet membrane, or the lipid bilayer. So lipid solubility plays a key role as well as molecular size. So smaller molecules that are more lipid soluble will freely pass through the lipid bilayer membrane. So the rate of diffusion, shown here as alpha, equals the available surface area plus the concentration gradient divided by the resistance of the membrane. That's based upon lipophilicity and molecular size, and the thickness of the membrane. So rate of diffusion can be calculated if these values are provided. So we now go back to the composite cell to try to piece this all together. So let's think of the cell membrane in its entirety. That would be the cytoplasmic membrane, if we want to call it that surrounds the cell or contains the cell. So a tin can is the container within which you find your pea soup. So it contains the pea soup. It holds it into one place. So there's going to be an apical, a basal, and a lateral cell membrane, and we'll see this in a cell in just a moment. There can be apical modifications. We saw that on the previous composite cell, like microvilli, to increase the surface area for absorption or passage of materials. They can be ciliated, so it'll sweep substances along a track. It contains cytoskeletal elements. We saw that already, attachments of them at least. The cilia do contain the cytoskeletal elements for movement. It is a mobile or motile projection. So here we see an example of a cell, an apical membrane here at the top of the cell. This is a typical epithelial cell, so this would be a brush border, say, of a kidney cell exposed to a surface lining. In this case, the lumen of a part of the urinary tube, the nephron. And then on the opposite side, where it would be interfacing with the underlying connective tissue matrix, the basal cell. So right now, from our preview of functional membrane proteins, we know that we would have many of these membrane proteins in the basal membrane that would be interacting with the extracellular matrix proteins, as we had illustrated. We probably would have many, many transport proteins in the apical membrane, but if we're going to resorb something from the urine, we would bring it from the lumen across this membrane through the cell and then pump it back out of the cell on the opposing side. And then these, of course, would be the lateral membranes. It's actually around the circumference of the cell. So here's an example. It's an illustration of ciliated pseudostratified columnar epithelium, the respiratory epithelium, and we are illustrating the motile projections. These are long projections, the cilia, and of course they move to move mucus through the respiratory system. What about the cytoplasm now? So it is the fluid portion that contains the cytosol, the clear fluid. So that's the other name for the intracellular fluid itself. The cytoplasm includes the cytosol with structures suspended within it, the organelles, especially the membrane-bound ones, and there are other structural organelles like the cytoskeleton. So let's take a moment to discuss the cytoskeleton. There are many proteins that make up the cytoskeleton. In this panel in the upper right, we see an illustration of immunofluorescence, labeling the nucleus in blue and various cytoskeletal elements in green or in yellow. So we see the cytoskeleton extends with the cytoplasmic membrane and obviously contributing to the shape of these cells. So what are some of these proteins? Actin is a major one. The intermediate filament is referred to as a guy wire. These are permanent cytoskeletal proteins. Microtubules are another, so they're always present, but they're made and broken down on an ongoing basis. So once you lay down an intermediate filament, it stays there for it's permanent, basically. A microtubule is assembled and disassembled on an ongoing basis. They are stiff tubules. They contribute significantly to the shape of the cell, and as we know, they to the intracellular transport or movement of organelles. So the gross intracellular movement, and they're found throughout the cell. So the microfilaments are next. They are meshwork of tiny rods. They're found just inside the cytoplasmic membrane. So we saw that already. Another example of microtubular associated intracellular movement is the spindle apparatus that forms during cell division. And we'll just move on to look at an illustration of these three types of cytoskeletal fibers. So the microfilament is shown on the top here on the right, and we see the multiple actin subunits, and it has a diameter of 7 nanometers. The intermediate filament is shown here. And there are fibrous protein subunits that are assembled and they spiral together, so it's a very complex multi-unit protein, and it produces a 10 nanometer diameter, whereas the microtubule has tubulin subunits. They are put together and assembled in such a way that they spiral. They're actually hollow, therefore tubules. Small tubes or tubules, and they produce overall a 25 nanometer diameter. So we see these intermediate filaments very close to the cytoplasmic membrane, the microfilaments, and the intermediate filaments spanning away from those, projecting in, and then the microtubules extending throughout the entire internal aspect of the cell. Here's an illustration, say, of an endothelial cell, because we can see it's a kind of a thin, flat cell, showing the cytoplasmic membrane on the outside and the inside of the cell. Say this is exposed to plasma. Plasma membrane is another name for cytoplasmic membrane, and this would be facing the interstitial fluid. So we see the microfilaments are little strips along the inside of the membrane, and the intermediate filaments, the guy wires, are connecting from the cell membrane to the microtubules, and then the microtubules move throughout, and the intermediate and the intermediate filaments and the microtubules contribute or coordinate movement of the intracellular molecules. Here we see endoplasmic reticulum, so their placement and any type of movement. This slide is illustrating the distribution of the cytoskeletal components, similar to what we just saw in the black and white illustration. So the microfilaments, we see their thick strands, collective bundles running along the extension of the cytoplasmic membrane. The intermediate filaments shown here, scattered throughout, forming this network mesh, such as illustrated here and we saw previously. And then the microtubules, we can see these very long strands almost make out the individual strands. So the microtubules are associated with movement within inside the cell, such as for an organelle. So they do interact with the motor molecules. And here we see a motor molecule with two little legs that interact with it, and with the expenditure of ATP, the organelle bound to the receptor on the motor molecule is going to be moved along the microtubule. In panel B, we see an interaction between two microtubules where the motor units, the motor molecules, are moving one microtubule with reference to another. The best example of this illustrative process, of course, is the sliding filament theory for muscular contraction. Here we see the cilia with its cytoskeleton components internally, and of course those are key for producing the movements such that the fluid on the surface, in this case external environment, can be moved. And then of course the other example we gave for microtubules was the spindle apparatus. Here we see, of course, the centrioles, as illustrated here in higher magnification, having a similar arrangement of microtubules, not exactly the same, but we're just passing through this in a cursory fashion. So intracellular movement and shape changes associated with cell division. Here we see this as in metaphase. The microvilli, on the other hand, are not motile, but they do contain cytoskeleton. So here we see a scanning electron micrograph of a microvillus on the apical membrane of cells. These are intestinal cells, the epithelial cells. And here we see in the very tip, there's a cap of proteins attached to the end of the microfilaments. And then we see the actin microfilaments running the entire length of each microvillus, and they support it. And there are cross links for the actin binding proteins. Where the actin binding proteins link those microfilaments together, and of course to the plasma membrane. And then these are interfaced with the intermediate filaments just subjacent to the apical membrane. And here we see an illustration of the microfilaments for support. We start first with orienting to the lipid bilayer of the membrane. We see another form of illustration of the proteins. It's filamentous and then forms an alpha helix for the transmembrane spanning segment, and forms an intracellular loop with another transmembrane spanning segment. And we must have a third one there someplace with finally an intracellular tail. So we have an extracellular tail, extracellular loop, intracellular loop, and an intracellular tail on this. Here we see complexing of a couple of these, a single one or another peripheral protein that goes in and doesn't become exposed. It doesn't to the outside. It doesn't span the complete length of the membrane. So we see the polar heads and the nonpolar tails of the membrane. And then this slide otherwise is showing that the microfilaments form the backbone of the microvilli that increase the surface area of some cells, such as the intestinal cells that absorb nutrients. That was the previous slide, excuse me. In this slide, panel B, we're seeing the actin filaments along with the ankyrin and spectrin proteins that support the donut shape of the red blood cells. So here we see ankyrin and spectrin, and then the actin suspended here that can draw in the cell membrane of the center of a red blood cell, so it is concave, biconcave disc, kind of like a throat lozenge. So now we can discuss the nucleus. It is a large sphere, as we had seen. It's enclosed by a double-layered nuclear membrane, 2 lipid bilayers, and there are nuclear pores in that nuclear membrane that allow for the movement of molecules into and out of the nucleus. It contains nucleoplasm, the term that I couldn't recall in that previous slide, which is the fluid portion within the nucleus. And then there are formed structures, such as the nucleolus, which is made up of ribosomal. It's the site of ribosomal RNA synthesis, so there are many proteins there, and a lot of RNA, and of course the DNA that contains the genes for the ribosomal, encoding the ribosomal RNA. And of course there is DNA that forms the chromatin within the nucleus, and it is obviously, as we know, used as the genetic template. So here we can see the nuclear envelope surrounding and containing the nucleus with the nucleolus here. So the nucleoplasm and the chromatin would be dispersed within. And we can see that there is an intermembranous space between the 2 lipid bilayers of the nuclear envelope. Intermembranous because there's 2 lipid bilayer membranes making up the envelope. I always use the metaphor of one wiffle ball inside another wiffle ball. You perfectly align the holes in each wiffle ball and then kind of insert a little plastic tube to attach those 2 aligned holes, the inner and the outer wiffle ball. The wiffle balls are the lipid bilayer membranes. And then the little plastic tube you shove through the holes is the nuclear pore guard proteins, and we'll see those in a little bit higher magnification in a moment, I believe. Here we see endoplasmic reticulum being continuous with the external membrane of the nuclear envelope. So the synthesis of a protein that would end up in the nuclear membrane would occur here on the the rough endoplasmic reticulum membrane, and then that protein would simply migrate, so fluid mosaic model, in here, find where it's supposed to be. If it has to go to the inner membrane, it'll push through at a nuclear pore, and then find its way onto the inner membrane. So it's really quite phenomenal. If you have a chance to read an article on that, any of the nuclear envelope proteins, you should really enjoy it. So here we see that same thing at a slightly higher illustration, and we can see the actual nuclear pores in this surface view of a sputter-coated. They sputter gold onto the surface of the nucleus, and you can see the nuclear pores. So what is the process of utilizing the genetic template? So we have three processes: transcription, translation, post-translational processing. There's also RNA processing to be put in here. So DNA to RNA is transcribing a code into a message. Let's think of messenger RNA to push this example. The message has to be processed. The initial message that was the initial transcript RNA has to be processed until you have, so that you have an RNA that can be used to be translated into a protein, and then the protein has to be processed so that you get a functional protein. So we will look at this protein synthesis in more detail in a subsequent lecture.